So how does this proofreading exonuclease work? Let's go here. So I've illustrated the reaction above or over here. Can anybody tell me, is it just reversing the synthesis reaction? Because that's one way you could think it could work. You could just do the reaction in reverse, right? So give me a reason why it can't do it in reverse.
We see that what comes off is the deoxynucleotide monophosphate.
And what would it be if it was reversing it? It'd be a triphosphate. And what would be at the top?
We have 2 inorganic phosphates.
Yeah, either 2 phosphates, or if we're lucky, we might find a pyrophosphate around, okay? So it's not the reverse reaction, and it can't be for the reason that I told you before, that essentially the synthesis reaction is irreversible once you take those 2 phosphates apart, okay? The enzyme could have been clever, kept a pyrophosphate around long enough to see whether it was correct after it made the synthesis, but that's actually probably pretty difficult for it to do, okay, for the reasons we talked about before. So how does it do this instead? So it uses a separate enzyme. So okay, and these proofreading exonucleases are, so first of all, an exonuclease deletes, or degrades is what I should say, from one end. Does anybody know what the alternative to an exonuclease is? What it's called? Probably most of you used one. What are the things that you cut DNA with when you're cloning? Restriction, what kind of nucleases? Endonucleases. So endonucleases cut inside a piece of DNA. Exonucleases need an end to act. And typically these just chew away a little bit of the end of the DNA. Okay? A proofreading exonuclease acts in a 3' to 5' direction. Okay? And in this case, we don't have to worry about what we're talking about, which direction, because the only thing to act on is a piece of DNA, and the 3' end, and it doesn't matter which end of the DNA, I mean, which part of the double-stranded DNA is around, as long as there's a 3' end, these types of exonucleases can act on them. Okay? So it's a 3' to 5' exonuclease, which is the opposite direction, right? It's degrading from the 3' end instead of extending from the 3' end as DNA polymerase is. Okay? So how does this work? How does it know what's going on, and how does it get access to the DNA it needs to act on? So as we just said, the reaction slows when you have a mismatch. Okay? And it also is true that when you have a mismatch, you already have a frayed end of the DNA, right? The last base pair at a minimum is not base paired. Okay? So what that means is that and I should point out that these are typically part of the same polypeptide as the polymerase. Okay, so when there's a proofreading exonuclease, it's almost always part of a dienzyme polypeptide. That is, there's a synthesis part of the polypeptide, and there's a exonuclease part of the peptide. And what happens is that the frayed or non-base paired has low affinity for the polymerase active site, okay? But it has the single-stranded DNA end of the primer okay, or the 3' OH, and has high affinity for the exonuclease site. I should say active site. Okay, and I'm just going to show you what happens then. So you get a mismatch right here. It's falling apart at the ends. It's not a good substrate for the polymerase, but this single-stranded region will basically come out of the active site and move to this base of the palm is typically where the active site for the exonuclease is, and typically 1 to 3 nucleotides will be taken off. So very often you remove more than just the mismatch. You remove a couple extra nucleotides, and that's fine because you'd rather remove the wrong one and a couple of right ones, redo it and get them all right, than to just have to worry about, oh, how much am I going to resynthesize? You'll find the more you study biology, the more sort of biology would rather be overworked but correct than underworked and incorrect. Okay? So doing a little extra work to make sure it's right is worth doing. Once you actually cut that away, then this single-stranded DNA will reanneal correctly with the entire template, and it will shift until you have the 3' OH in the appropriate position for continuing synthesis. Okay? So basically, this frayed end turns out to be a good substrate for the exonuclease, but when you remove that, then you have a fully base pairing double-stranded region. It will reanneal rapidly, okay? And then it will go back to the normal synthesis reaction. Right. So the one thing I didn't say about the repair is how much it helps. So basically, having a proofreading exonuclease on a polymerase gives you about 100-fold more accuracy. Okay? So you'll have one mistake in 10 to the 7th base pairs synthesized. Okay, so that's pretty good. We're now only 3 orders of magnitude off the final amount. And basically in, I believe, lectures 5, 6, and 7, we'll talk about how the DNA repair mechanisms work. And that's what gets you from 10 to the 7th to 10 to the 10th. So in particular, mismatch repair. Basically, if for some reason you have a mistake, you continue synthesizing, you somehow don't stop long enough to do the proofreading exonuclease. If you basically end up with a mismatch somewhere in here, once you get down to here, the polymerase really can't tell anymore, and it just keeps going. Okay? But there's a whole separate machinery that goes around the genome and looks for mismatches. And in fact, it's targeted in such a way, and this is the really special part of this, that it will take the wrong base, identify it, and remove it. Because you can imagine, once you have a mismatch, you don't actually know a priori whether the C next to a T, whether the C was right or the T was right initially. And there are ways the cell knows how to do that, and we'll talk about how that works.